Hello, everyone, and welcome to this first of two screencasts on the digestive system. Now, if you look at the opening slide here of our PowerPoint, it says the digestive system and body metabolism. So there's a lot for us to cover here. So what I'm going to do is I'm going to split these into two separate PowerPoints. So we're going to talk about the digestive system first. This is going to be one of two PowerPoints on the digestive system. And then we'll talk about body metabolism, and that there will be two lecture screencasts on body metabolism. So the way we're going to divide it out here is in this first section, we're going to be taking a look mainly at the functional anatomy of the digestive system. Then whenever we get into body metabolism, we'll be talking more about physiology, about how things get done, how does the digestive system do what it does, and how are nutrients metabolized once they're absorbed by the body. So let's go ahead and get started. And I'm going to be moving fairly quickly through this, so if you need me to slow down, just hit the pause button whenever you need to. So the digestive system and body metabolism, we take a look at it. The question becomes, what is digestion? Simply put, digestion is the breakdown of ingested food. And a second part of the process then is once the food is broken down, it has to be absorbed. The nutrients from the breakdown of the food have to be absorbed into the blood. Metabolism, on the other hand, is the production of cellular energy. We're talking ATP here, mainly through the process of cellular respiration. That's pretty much as I'm going to say about metabolism up to this point in time. We'll save the rest of it for our PowerPoint on metabolism after we complete the digestive system. But metabolism is broken down into both constructive and degradative cellular activities. In other words, metabolism can include building things and it can include breaking things down. And again, we'll take a look at that further in our lecture on metabolism. Okay, so the rest of this PowerPoint is going to be about the digestive system. So what are the organs of the digestive system? Well, whenever we take a look at these organs, they're going to be subdivided into two main groups. And we'll look at them from that perspective. The first group are what we call the alimentary canal organs, or simply the alimentary canal. This is a continuous coiled hollow tube. And, clinically, the alimentary canal will be referred to as the gastrointestinal tract or the GI tract. So it's kind of one of the same. In anatomy, we like to use the term alimentary canal. In medicine, we like to use the term gastrointestinal or GI tract. In addition to the alimentary canal, though, there are also what we call the accessory digestive organs. The accessory digestive organs are ones that are involved in digestion, but the food doesn't actually pass through them. So here is our slide where we kind of put all the organs of the digestive system together. We can see, and we'll be referring back to this, all the organs and their relationships to one another in the body. All right, let's begin with the alimentary canal organs. So the alimentary canal organs would include the mouth, the pharynx, the esophagus, the stomach, the small intestine, and the large intestine. And again, these organs are all organs through which the food is actually going to pass through during the digestive process. And of course, at the end of the large intestine there, we see the anus as well, the opening to the outside. All right, so we're going to take these one by one. Let's take a look first of all at the mouth, which includes the oral cavity. So let's take a look at the structural anatomy of the mouth. First of all, we have the lips, or from the Latin, the labia. These protect the anterior opening to the mouth. Next up, we have the cheeks. The cheeks form the lateral walls of the mouth. The hard palate forms the anterior roof of the mouth, and it's formed by the palatine bones and the maxillary processes of the maxillae. Next up, we have the soft palate. It forms the posterior portion of the roof. So if you kind of run your tongue from the front to the back, you'll notice you have the hard portion of the roof of your mouth, and then it becomes soft, and that's because there's no bony support underneath of it, and that's the soft palate. The soft palate then will end in a fleshy projection that extends in a downward fashion. We oftentimes refer to it in lay terms as the gumdrop, but it's known as the uvula. Next up, we have the vestibule. The vestibule is described as the space between your lips externally and your teeth and gums internally. So you can kind of put your tongue between your lips, and you can put them between your teeth. And when you do that, you're putting your tongue into the vestibule. The oral cavity itself is the area of the mouth that is contained by the teeth. So everything, take your tongue and run it around the edge of your teeth. Everything inside the teeth is referred to as the oral cavity. And then we have the tongue, and the tongue is attached to the hyoid bone and to the styloid processes of the skull, and also by the lingual frenulum. So if you lift up your tongue and kind of look into the mirror, you'll see kind of a line of mucous membrane there that attaches the tongue to the floor of the mouth, and that is the lingual frenulum. And you may recall from A&P 1 where you had to learn the structures of the skull and these little bony projections back here. The styloid processes will kind of look like little pen or pencil points. You might wonder why you need to learn those. Well, now we know that the tongue is actually attached to those styloid processes along with the hyoid bone. Okay, next up, we take a look at the tonsils, and we've talked about the tonsils before. So remember, we have 3 sets of tonsils. 2 sets we talk about here, the palatine tonsils and the lingual tonsils. Remember, the palatine tonsils are those ones that look like golf balls on either side of the pharynx. And the lingual tonsil is the one that's back on the surface of the tongue. You can see how far back it is around the curve of the tongue. So it's very difficult to see. And again, we've already kind of talked about the functional anatomy of those tonsils as well. Okay, so what happens in the mouth? What are the processes of digestion that can occur in the mouth? Well, generally speaking, we have the mastication or chewing of food. So chewing is referred to as mastication. And in addition, while the food is being chewed, we're going to mix it with saliva. So a second process is the mixing of that masticated food with saliva. And then finally, whenever we're ready, how many chews do you take before you swallow your food? 10, 20 chews, 3 chews, or you just swallow in one big lump? That's not good or healthy for you. But once you're ready for it, voluntarily, you initiate swallowing by lifting up your tongue. So initiation of swallowing by the tongue is another process of the mouth. And of course, the mouth, we studied this way back at the beginning of the semester whenever we were taking a look at the special senses, also allows for the sense of taste via the taste buds. From the mouth, we move on to the pharynx. So let's take a look at the anatomy of the pharynx. Remember, the pharynx is elongated, so we have 3 distinct regions of the pharynx. There's the nasopharynx, and actually the nasopharynx is not really part of the digestive system, but the other 2 regions are, and that would be the oropharynx. The oropharynx is behind the oral cavity, and the laryngopharynx, which is below the oropharynx and is connected to the esophagus. So here on the side diagram, we can see the 3 regions of the pharynx. From a standpoint of function, the pharynx serves as a passageway for both air and food, and as a result of that is part really of the digestive system and the respiratory system, as we've seen in our coverage of the respiratory system. So when food enters the pharynx, it is then propelled to the esophagus by 2 muscle layers. there is, and this is kind of backwards on this slide, there is an outer longitudinal layer, that should say outer longitudinal layer, and there is an inner circular layer of smooth muscle tissue. And these two layers of smooth muscle tissue uh, act by alternating contraction to move the food from the pharynx down into the esophagus and even beyond. These alternating contractions of these smooth muscle layers is referred to as peristalsis. Okay, moving on. Our next component is the esophagus. The esophagus runs from the pharynx to the stomach, and as it does, it passes through the diaphragm. It conducts food then again by peristalsis. Again, this is a slow rhythmic squeezing, alternating contraction of the layers of smooth muscle tissue. And the food is conducted by peristalsis down into the stomach. The esophagus, however, is a passageway for food only. The respiratory system branches off after the pharynx. So we know the respiratory system kind of picks up with the larynx at the top of the trachea. All right, so whenever we take a look at the alimentary canal organs, we look at how they're organized. And basically, whenever we look at the alimentary canal, it's a tube. We oftentimes refer to it simply as a food tube. And that tube, like other tubes, think of arteries and veins, they have layers that make up the wall of the tube. So we're going to take a look at these layers now. The first layer we'll look at is the mucosa. This is the innermost layer. The mucosa is a moist membrane. And the mucosa is made up of several subcomponents. The first is a layer of surface epithelium. So there's an epithelial layer. Remember what epithelial tissue is. And then beneath that, there is a small amount of connective tissue. And that small amount of connective tissue is referred to as the lamina propria. And then below that, there's a small layer of smooth muscle tissue. So all three of these components make up the moist membrane that we refer to as the mucosa. Mucosa contains surface epithelium, a small amount of connective tissue called the lamina propria, and a very thin, smooth muscle layer. Next, as we move out from the mucosa, we move to the submucosa. The submucosa lies just beneath the mucosa. Now, you have to kind of be careful about this because when you think of beneath, you think of below. But remember, we're talking about a circular tube here. And so the mucosa is the innermost part of that circular tube. We'll show you that in the diagram as we move forward. But then surrounding that is going to be the submucosa. So sub technically means below, but it's going to be surrounding the mucosa. And we say it's technically just beneath the mucosa as we move out from the lumen of the alimentary canal organ. The submucosa consists of soft connective tissue. Here's where we're going to find a lot of blood vessels, nerve endings, and lymphatics that are going to support the alimentary canal organ. Next up, we have the muscularis externa. This is made up of smooth muscle tissue. There is an inner circular layer. Now we have it right on this slide. The circular layer is the inner layer. And there is an outer layer, which is referred to as the longitudinal layer. And we'll see how that works again whenever we take a look at the diagram. And then the final layer of the alimentary canal organ is the serosa. This is the serosa. Think of serous membranes. Serous membranes have what? They have, two layers. One layer is attached right to the surface of the internal body organ. The other layer doubles back and attaches to the body wall. So the serosa, the outermost layer of the serosa, is referred to as the visceral peritoneum. This is the portion of the serous membrane that is attached right to the visceral organ, right? And the visceral organ would be the alimentary canal organ inside the body. So this is the outermost layer, and it's referred to as the visceral, and this time peritoneum. Then there's a layer of serous fluid producing cells. So just as we saw, let's say, for example, with the heart, where we saw the pericardial sac, serous membrane, and with the lungs, the pleura, there's always that inner layer, the visceral layer, and then there is an outer layer, the parietal layer, and between the two layers, there is going to be fluid, serous fluid, and that's produced by the serous producing cells. Okay, so we didn't have in that previous slide about the parietal peritoneum. That's the portion of the serous membrane that doubles back on itself and attaches to the body wall. So now let's take a look at this slide, this idealized artist rendition of an alimentary canal organ. So what we're going to see here is in here, we'll start right here. This is the lumen of the organ. This would be where the ingested food is going to pass. And then surrounding the lumen, we're going to see this layer of columnar epithelial cells. Okay, and that layer of columnar epithelial cells makes up the surface epithelium. Then we see a thin layer of connective tissue, the lamina propria, and then we see a thin, smooth muscular layer. You can tell these are smooth muscles. You take a look, you can see the spindle fibers that are there. Okay, these three components, then, the surface epithelium, the simple columnar epithelium, the lamina propria, and the thin, smooth muscle layer make up the first layer of the alimentary canal organ, the mucosa. Moving out from the mucosa, then, this is where we have that submucosa, and you can see lots of blood vessels, arteries, veins, glands. We can see also nerves here, and there are also lymphatics that are in there as well. And these are all found in the submucosa. Moving out from the submucosa, then we have the muscularis externa, and it has two layers, longitudinal muscle layer and a circular muscle layer. Now the longitudinal layer is the outermost layer. And if you take a look at it, you can see why it's called the longitudinal layer, because look at how the spindles are organized lengthwise along the length of the organ. And the circular layer, look at how the spindles are organized around the diameter of the organ. And so the circular layer is the inner layer, and the longitudinal layer is the outer layer. Alternating contractions of these layers will move food along through the alimentary canal. All right, then finally we have out here, we have the visceral peritoneum, the outermost layer, the serosa, and we can see the visceral peritoneum here. And notice how the visceral peritoneum, if you come around, it comes all the way around the organ. And here we'll pick it up down here. It comes around the organ together. And then what's going to happen is it's going to come together here and form a structure called the mesentery. Whenever you take a look at the cat dissection, you'll see the mesentery, and we'll take a look at that in that dissection. And when you see the mesentery, this is how the mesentery forms. It kind of has like two layers of bread with the blood vessels kind of in between. It contains the blood vessels, the nerves, and the lymphatics. And then what's going to happen is this mesentery will kind of fold back on itself to attach to the body wall on either side. So out here, not shown, is where we would have the parietal peritoneum. Okay, so I hope that helps to make it a little more clear the structural organization of the alimentary canal organs. And they all have that basic, that same basic organization. If there are some differences, we call them modifications, and we'll take a look at them as we move along. So now let's take a look at the stomach. Let's move on from the esophagus to the stomach. Stomach is located on the left side of the abdominopelvic, or the abdominal cavity, I should say. Food is going to enter the stomach at a sphincter. A sphincter is a circular piece of muscle tissue that kind of acts as a valve. So whenever we swallow the food, it's going to pass down from the pharynx into the esophagus, and eventually it's going to arrive at the top of the stomach. At the top of the stomach, there's a valve, and that valve is closed. It's called the cardioesophageal sphincter, circular piece of muscle tissue. When the food comes in contact with that, it's going to open up and it's going to allow the food to pass down into the stomach. So we say the food enters the stomach through the cardioesophageal sphincter, or sometimes simply just the cardiac sphincter. Now, whenever we take a look at the stomach, it's a sac-like organ, so we're going to divide it into regions. The first region we take a look at is the cardiac region. The cardiac region is the portion of the stomach that's up near the heart, cardiac pertaining to heart. And within the cardiac region, we're going to find a dome-shaped part of the stomach. That dome-shaped part is referred to as the fundus. And then the mid portion of the stomach is referred to as the body of the stomach. And then at the opposite end of the cardiac region, we have a region called the pylorus. This is a funnel-shaped terminal end of the stomach where the food is going to leave the stomach and enter into the next portion of the alimentary canal, the small intestine. So food is going to empty into the small intestine at the pyloric sphincter. So just like there is a valve at the top of the stomach called the cardiac sphincter that has to open and allow the food to enter the stomach, there's also a valve at the terminal portion of the stomach, the pyloric sphincter, that allows food to exit the stomach after a period of time in the stomach and mixing and churning that occurs in the stomach. We'll talk about that more a little bit later on. So internally now, whenever we take a look at the stomach, what we're going to see is there are folds in that mucosa. So remember, the mucosa is the innermost layer of the wall of the alimentary organ. So the mucosa of the stomach is thrown into these internal folds, and these are called rugae. And this is what allows the stomach to expand and contract. So when the stomach is empty, what will happen is the stomach will collapse sort of into these folds of mucosa called rugae. So if the stomach has a lot of food in it, it will distend, and these folds will kind of unfold or disappear. You wouldn't see them in a full stomach that's greatly distended. But when the stomach is empty, these folds will form, and you would see these internal folds of mucosa much more easily. Now, externally, whenever we take a look at the stomach, we're kind of bopping around here, but that's okay. The stomach is kind of like a J-shaped sac. And because of that, we see two curves that we talk about. One is called the lesser curvature. The lesser curvature is the inside smaller curve of the stomach, and there is a greater curvature, which is the outside larger curve of the stomach. We'll see that in the diagram in a moment. So the layers of the peritoneum that are attached to the stomach. So again, the stomach is going to have that kind of that outside serosa that forms the outer wall of the stomach. It's made up of a serous membrane, and that serous membrane attaches partly to the stomach, and then it kind of folds back on itself to attach to the body wall. But it also, as it does, it forms these two curtain-like structures. One is called the lesser omentum. The lesser omentum actually attaches the stomach to the liver at the lesser curvature. So it's kind of a connection between the lesser curvature of the stomach and the liver. The greater omentum attaches the greater curvature of the stomach to the posterior body wall. And whenever you take a look at the cat dissection and you look at the greater omentum, you'll see that you can actually lift it up, and it truly does kind of look like a curtain. It contains a lot of fat, and that fat is important because it helps to insulate the body organs, protect against uh, uh, great temperature differences, helps to keep the temperature regulated. It acts as a cushion. Think about it. With the stomach, it's below the rib cage, so there's not a whole lot of bony support to protect the stomach. So it provides cushioning to help protect the stomach. And it also helps because it forms a curtain down over other organs. It also helps to provide protection for other abdominal organs like the small and large intestine. So here we go. We'll put it all together with this diagram of the stomach. So up here we can see the esophagus. It's not real clear the ring of smooth muscle tissue here that forms the cardioesophageal sphincter or the cardiac sphincter. But this does act as a valve. So when food comes down through the esophagus, wants to enter the stomach, that muscle tissue has to relax. The valve opens up and allows the food to drop into the stomach. This upper let me just go back to that. I apologize for that. This upper region of the stomach is the cardiac region of the stomach. And notice this dome-shaped area in the cardiac region. This is referred to as the fundus. And then we come down here. This would be the body of the stomach. So you have to watch out because it looks like this is pointing to these two muscle layers, but it's not. It's really just kind of pointing generally to this mid portion of the stomach. And this mid portion of the stomach is referred to as the body of the stomach. And then we come down here to the lowermost terminal portion of the stomach. You can see how it kind of forms a funnel-shaped region here. And this funnel-shaped region is referred to as the pylorus. Now, I do have to mention here that you want to make a correction on your slide. You see where it says pylorus, and it points to this area right here. That would be incorrect because this would be into the duodenum after the pyloric sphincter. So this line should really point over into this region here to point out the pylorus, the terminal region of the stomach. So I hope you got that. And then this area right here, this would be the pyloric sphincter, again, that ring-shaped valve that's formed from muscle tissue. And this has to open up to let digested food, kind of a liquefied mix we called chyme, to move out of the stomach and enter into the first part of the small intestine, the duodenum. Okay, moving over here, we take a look at, see the kind of J-shaped curve of the stomach. And as a result, we can see this large outer curve, which is referred to as the greater curvature. And then we can see this smaller curve right here, the lesser curvature. Remember, the lesser omentum is attached to the lesser curvature and attaches the stomach to the liver above. And the greater curvature here, the greater omentum is attached to that, and it hangs down from the stomach kind of like a curtain. And then we have the last but not least, we'll talk about the muscularis externa. And whenever we take a look at the muscularis externa, we're going to see a modification here in the stomach. There are going to be 3 layers instead of 2. So there is a longitudinal layer, there is a circular layer, and now we're going to have a modification to a 3rd layer, the oblique layer. You can see here in this oblique layer that we're trying to show that the muscle fibers, the spindles, are oriented in kind of a diagonal fashion. This helps with mixing and churning that happens in the stomach. So basically what you're going to have is when food enters the stomach's going to be able to move this way to swish food. The circular smooth muscle tissue is going to have it move this way, and then obliquely it's going to be able to contract this way. And this is going to create a lot of mixing and churning to help with the digestion process in the stomach. All right, and speaking of the digestive processes, what are the stomach functions? Well, first, the stomach is going to act as a storage tank for food. So what we're going to see is that food can be stored in the stomach depending upon the size of the meal for as much as anywhere from 4 to 6 hours. It's also going to be the site of food breakdown. So when food enters the stomach, it's going to be mixed and churned, lots of water, lots of acid in there, lots of enzymes that are going to help to start to break the food down. And so in the stomach, the chemical digestion of protein is going to begin because there are going to be enzymes that are going to be injected into the stomach that are going to cause protein breakdown. So after a period of time that the food is stored in the stomach, again, 4 to 6 hours, the stomach will deliver the chyme through the pyloric valve. The processed food is kind of a liquefied processed food now, and it'll deliver it into the small intestine. All right, let's take a look at specializations from our basic anatomy of the alimentary canal organs. In the stomach, we're going to see a layer of simple columnar epithelium. This simple columnar epithelium contains some specialized cells. One of the specialized cell types are what we call mucous neck cells. These mucous neck cells produce a sticky alkaline mucus. Remember what alkaline means? With a pH, the mucus has a pH above 7. In addition, there are going to be in the simple columnar epithelium gastric glands. These gastric glands will secrete gastric juice. The gastric glands contain chief cells. These chief cells produce the protein digesting enzymes, which are referred to as, here we have in this slide called pepsinogens. They're actually, the enzymes are pepsins. Pepsinogen is the precursor to pepsin. So pepsinogen is initially produced, and then we'll see in the process of how the pepsinogen gets converted into pepsin once it enters the lumen of the stomach and then causes the breakdown, the digestion of the protein in the stomach. But the chief cells produce these pepsinogens that ultimately will become activated pepsins to digest protein. In addition, in the gastric glands, we're also going to find parietal cells. The parietal cells produce the hydrochloric acid that we hear so much about associated with the stomach. And then finally, there are also endocrine glands, and these endocrine glands produce a hormone called gastrin, which is very important in appetite and appetite control. Okay. Continuing on, so we take a look at those gastric glands. They're at the base of structures called gastric pits. These gastric pits are formed by folded mucosa. Now, not the rugae that we talked about earlier, but kind of inward folds of the mucosa. These gastric pits are part of glands and specialize, or I guess I should say lead to glands and specialized cells that are in the gastric gland region. So here's what we're talking about here. I think this will kind of help clarify things. We look over here at this little diagram of the stomach. So we're taking a look at the layers of the stomach wall right here. And we're blowing this up really big. So this area right here would be the mucosa. And in the mucosa, we're going to have these openings, which are referred to as the gastric pits. So here we can see the inward folds of the mucosa that form the gastric pits. And we can see the surface epithelium here. Notice the columnar cells here. Then at the base of the pit, we have the gastric gland. Okay, and we can see the various cell types in the gastric glands. And first, up here toward the top, notice we have a high concentration of mucous neck cells. These produce the sticky alkaline mucus. Okay, now embedded down below here we have parietal cells. And the parietal cells, remember, if we look back to our previous slide, the parietal cells, I guess it's even one more. Let's go back here. The parietal cells produce the hydrochloric acid, while chief cells produce the protein digesting enzymes. So keep that in mind. That's going to be important. This organization is going to be important as we go forward. Okay, so let's get back to where we were. So here are the parietal cells, right? And they're going to produce the hydrochloric acid. So here's a parietal cell here now producing hydrochloric acid. And once produced, the hydrochloric acid is going to move up through the gland and into the lumen of the stomach. Some more mucous neck cells that we see here. And then we're also going to see down here these purple chief cells. Now remember, these purple chief cells are going to produce the pepsinogens. Okay, so here we can see a chief cell, and that chief cell is going to produce pepsinogen, and it's going to be released to the internal portion of the gland, and then it's going to move up into the lumen of the stomach. Now the importance of this is this. We do not want to produce pepsins down here that will digest enzymes, because the enzymes, if they're activated here in the gland, will start to digest the proteins that make up the cells. We don't want it, and that would destroy the cells. We don't want that to happen. So we don't want the protein to become active until it moves into the lumen. So once the pepsinogen moves into the lumen, notice the hydrochloric acid was produced by the parietal cells above. Now the pepsinogen is going to the inactive protein digesting enzyme, is going to interact with hydrochloric acid, and the hydrochloric acid is going to convert the pepsinogen into pepsin. Once formed, pepsin will also be involved in conversion of more pepsinogen into even more pepsin. This ensures that the enzyme isn't active until it's in the lumen of the stomach, where we want to digest that protein from the food that we ingested. Okay, in the lumen, with all this additional mucus, we have a thick layer of mucus here now that's lining the lumen, and that mucus protects the cells below from the action of the hydrochloric acid and now the pepsin. All right, let's move on to the small intestine now. The small intestine is your body's major digestive organ, and it's also the site of nutrient absorption into the blood. So this is where the majority of digestion is going to take place, is in the small intestine. And once the food is digested, it is also going to be absorbed. Okay, I apologize for getting a phone call during this, but sometimes those things happen. Whenever we take a look at the small intestine, basically it's a muscular tube. It's going to extend from, that should say from, not form. It's going to extend from the pyloric sphincter at the end of the terminal end of the stomach to another sphincter called the ileocecal valve, which we'll take a look at in just a bit. The small intestine is suspended from the posterior abdominal wall by that mesentery that we talked about earlier. Remember, part of the serosa. The small intestine is fairly long, and so we're going to divide it into different regions. Let's take a look at those different regions. We have the duodenum first. This is the portion of the small intestine that is attached to the stomach. It's going to curve around the head of the pancreas. We'll show you a diagram of that in just a bit. And then it will extend down to the next subdivision of the small intestine, which is the jejunum. Now, if you look, this is an indistinct subdivision. It's no place where you look at the small intestine and go, oh, I can see the duodenum ends here and the jejunum begins here. There's no landmark to look for. It's just generally where gradually the function of the small intestine changes. But the duodenum then, we might say, becomes the jejunum. So the jejunum is going to be attached anteriorly or above to the duodenum, and then it's going to be attached posteriorly or even below to the ileum. So the ileum, the third subdivision of the small intestine, will extend from the jejunum then on to the next division of the alimentary canal, the large intestine. All right, so let's take a look at chemical digestion in the small intestine. Remember, the bulk of digestion takes place in the small intestine. So whenever we take a look at this, the chemical digestion in the small intestine, and that means the breakdown of substances into completely different substances. So chemical digestion, let's say, for example, we're going to break down proteins into amino acids. We're going to break down complex carbohydrates like starches into glucose. And we're going to break down fats into fatty acids and glycerols. What's the source of enzyme for this chemical digestion to occur in the small intestine? And these enzymes, once produced, are going to enter the small intestine and they're going to mix in with the chyme, that liquefied food that's going to come out of the stomach. Well, some of the enzymes are produced by cells that are actually in the walls of the small intestine itself that we call the intestinal cells. But most of the enzymes are produced actually by an accessory gland, the pancreas. In addition, bile will also enter from the gallbladder into the small intestine. So as we take a look at it here, so here we can see, here is the duodenum coming out of the stomach. So the stomach would be up in this area right here in the pyloric region and pylorus. So we're picking up just after the pylorus and we're coming around this large portion of the pancreas. The yellow is the pancreas. So this would be referred to as the head of the pancreas. And we can see some ducts that are entering into the small intestine here. These ducts, the accessory pancreatic duct and the pancreatic duct, will carry enzymes produced by the pancreas and release those enzymes into the duodenum in this region, which is called the hepatopancreatic ampulla. And there's a sphincter, a valve that controls it right there, ampulla and sphincter, or what is sometimes referred to as the ampulla of Vater, V-A-T-E-R. Bile is going to enter into the small intestine at the same point, but the bile is going to enter as it's produced by the liver, or stored bile is going to enter that is released from the gallbladder via the common bile duct right here, and eventually in at the ampulla of Vater. Okay, next up, internally, when we look at the small intestine, we see a modification that the small intestine is thrown into a series of finger-like structures that are formed by the mucosa. These finger-like structures are referred to as villi, and their job is to give the small intestine more surface area upon which enzymes can act. So we can see here these folds in the small intestine, and then the folds on top of the folds. These finger-like projections or folds that we see here are villi. These help to increase the internal surface area of the mucosa. And that means the more surface area we have, the more area we have for enzymes to work, and the more area we have for the absorption of nutrients once the enzymes do their work. In addition to those villi, however, to increase the surface area for absorption, particularly even further, there are small projections in the plasma membrane of the columnar cells themselves. These small projections are referred to as microvilli. So here we can see some absorptive cells, and we can see here at the apical region of this cell that we can see how the membrane is thrown into these additional folds. These additional folds, these additional tiny finger-like projections are referred to as microvilli. And again, that increases the surface area through which nutrients can be absorbed, let's say from the lumen up here, once the enzymes act on them, can be absorbed through these cells and then passed into the blood. Okay, that moves us on to the large intestine. The large intestine is larger in diameter than the small intestine, and that's where it gets its name from. But we should note here that it's shorter than the small intestine. So the large intestine gets its name because it's larger in diameter, not because it's longer in length. The large intestine itself is going to form a framework around the internal abdomen and around the small intestine. And here we can see how it forms that framework around the small intestine and internally in the abdominal region. What are the functions of the large intestine? Well, one function of the large intestine is the absorption of water. Now, I got to pause here for just a second and say this about the absorption of water. A lot of people have a misconception that the large intestine reabsorbs the largest portion of the water. It does not. Water absorption also takes place in the small intestine. What happens in the large intestine, the absorption of water that occurs there, is kind of like tweaking the amount of water that has come out, so that as the water is resorbed and the food passes through, what's left after the nutrients are absorbed? Fecal matter. So that fecal matter will have a certain uh, uh, solidity to it, let's say. And if the fecal matter is too runny yet, then the large intestine will act to absorb additional water to get it to the right composition. If it's not uh, that runny, then the large intestine won't reabsorb so much water. In addition, the large intestine is responsible for the elimination of indigestible food from the body as fecal matter or feces or stool. Important to note here that the large intestine does not participate in the digestion of food. All the digestion takes place above the large intestine. The large intestine does have specialized cells in the mucosa. These are goblet cells. They produce mucus, and that mucus acts as a lubricant. If we were actually able to do the cat dissection, a lot of times we'll take your finger and move it along the large intestine. We open it up and you'd find that it's very slick inside. And that acts as a lubricant to help the feces move, or the stool move more easily through the large intestine. So let's take a look at a couple of components of the large intestine. First of all, we have the cecum. The cecum is the sac-like first part of the large intestine. Then we have the appendix. The appendix is an accumulation of lymphatic tissue, so it's going to have lymphatic function. It's going to contain those T cells and B cells. And sometimes the appendix can become inflamed, and when it does, that's referred to as appendicitis. The appendix is kind of like a finger-like projection of lymphatic tissue that hangs from the cecum. If we take a look at the cat, cats don't have a cecum, contrary to popular belief, because there are some YouTube videos out there that try to identify the appendix of the cat. Cat has a cecum, but it does not have an appendix. All right, other structures of the large intestine. So the large intestine is divided into regions. One region of the large intestine is the colon, and the colon is subdivided into an ascending colon, which runs up the right flank of the abdominopelvic wall. then a transverse colon, which runs across the abdominal wall just beneath the diaphragm. And then there's a descending colon, which runs basically down the left flank or the left side of the abdominal wall. The colon then ends as it forms an S-shaped curve called the sigmoid colon. And that's where the colon kind of curves back around to get inside the pelvic rim and then go down into the pelvis. So whenever we take a look at it, for example, a colonoscopy might be looking at the entire colon, and a sigmoidoscopy might be just looking at the sigmoid region of the colon. In addition to the colon, then, the next component of the large intestine is, in fact, the rectum. The rectum is kind of a storage area for fecal matter until the time at which we're going to defecate or release it. And then finally, the stool or the fecal matter is released through an external body opening called the anus. So these are all three components of the large intestine. All right, so there are some modifications of the muscularis externa of the large intestine that we'll take a look at now. First of all, the smooth muscle is reduced into three bands, and these three bands of smooth muscle tissue are referred to as the tenei coli. These muscle bands do have some degree of tone, slight contracted state, and because of that, and because the muscle doesn't go all the way around and only forms these three bands, whenever the bands contract, or because the tone of the bands, it kind of pulls the wall into these pocket-like sacs, and these pocket-like sacs are referred to as haustra. So back to our picture of the large intestine. So here we can see, here's the end of the small intestine's been removed, but here's where the small intestine ends. And at that ending, there is a valve that controls the release then of the indigestible material or feces into the large intestine. That's the ileocecal valve. Now notice the large intestine is larger in diameter than the small intestine, so there's this sac-like structure that kind of hangs from that attachment site, and that's referred to as the cecum. And then we can see this finger-like projection of lymphatic tissue, the appendix, which extends from the cecum. Remember, cats don't have that, or it's referred to as the vermiform appendix. Then the large intestine runs up the right flank. This portion that runs up the right flank is referred to as the ascending colon. And then there's a 90-degree bend here. And this bend occurs just underneath of the liver. So it's on the right side of the body, just underneath the liver. It's called the right colic flexure or the hepatic flexure. The large intestine then runs across the abdominal cavity just beneath the diaphragm. Then there is another kind of 90-degree bend in the colon, and this bend occurs underneath the spleen on the left side, so it's referred to as the left colic or splenic flexure. And then the large intestine runs down the left side of the body wall, the abdominal wall, and is referred to as the descending colon. Then we can see where it kind of forms this S-shaped turn that allows it to get back inside the pelvis. This is referred to as the sigmoid colon. And then that ends in the rectum, and then the anal canal and the external anal sphincter at the anal opening. Notice also here, we can see here at the top, we can see them pretty readily. These are those bands of smooth muscle tissue. So the smooth muscle tissue doesn't go all the way around. We just see it reduced into these bands, and these bands have a certain amount of tone to them. They're called the tenei coli. And because of their tone, look at how it pulls the wall into these sac-like structures. These sac-like structures are referred to as haustra. Haustra are normal. They're supposed to be there. Now, when we think about something like diverticulosis, what is diverticulosis and diverticulitis? So these haustra are normal sacs, but what can happen is because you have the lack of a muscle all the way around, it doesn't have quite the support. So what can happen is you can get out pockets from weakenings of the walls around the haustra, and these out pockets will be referred to as diverticula. So you can have, if you have these out pockets that form, you have what is called diverticulosis. If these out pockets become inflamed, then that is referred to as diverticulitis. Itis means inflammation. So difference between diverticulosis and diverticulitis. Just wanted to mention that before we move on. Okay. With that, we've gone through all the alimentary canal organs, the food tube, the GI tract through which the food actually moves. Now we'll take a look at the accessory digestive organs. Remember, the food doesn't actually pass through these organs, but they contribute to the digestive process. And so the first accessory digestive organ we have is the salivary glands. Then we have the teeth, the pancreas, the liver, and the gallbladder. So we'll be taking a look at each one of these individually. First of all, the salivary glands. Salivary means pertaining to what? Saliva. So they're saliva producing glands. We have three sets of salivary glands. We have the parotid glands, which are located here, just in front of the ears. An inflammation of the parotid glands due to viral infection is referred to as the mumps, by the way. Then we have submandibular glands, which are located here at the corner of the mandible. And these are referred to as the submandibular glands. And then we also have two glands here just underneath the tongue. And since they're underneath the tongue, they're referred to as sublingual glands. Now, all of these are ducted glands. So they are exocrine glands. They produce the mucus, and then the mucus is passed through the ducts and enters the oral cavity. The saliva itself is a mixture of mucus and also serous fluids. And again, it's kind of sticky, so it helps to, as we chew the food, form it into roughly an egg-shaped mass that we refer to as bolus. Saliva, however, also contains a enzyme called salivary amylase, and this enzyme is important in the digestion of starch. So starch digestion actually begins as we chew food and mix it with mucus in the mouth. The dissolved chemicals, once the food mixes with water and mucus and starts to partially digest, the dissolved and broken down chemicals then can attach to the taste buds, and then we get our various tastes. Okay, moving on to the teeth. The job of the teeth is simply to masticate or to chew the food up into smaller pieces. Humans actually have two sets of teeth, as you probably know, because you've all gone through this. The first set of teeth that we get are referred to as the deciduous or baby or milk teeth. They're called deciduous because they're going to fall out. So just like the trees out there that we have right now in the spring, they're all getting their leaves back on. But in the fall, they drop those leaves. Those trees that drop their leaves are referred to as deciduous trees. And then we have conifers that have their leaves or needles all year round. Just a little on the side there. But the same concept here, deciduous teeth, because they're going to fall out and they're going to be replaced by the permanent teeth. But as far as the deciduous teeth go, there are 20 teeth and that make up the deciduous teeth, and they're fully formed by the age of 2 or thereabout. However, as we mentioned, they will be replaced by a second set of teeth, the permanent teeth. The permanent teeth will begin to replace the deciduous teeth around the ages of 6 to 12. A full set of permanent teeth is 32, but some people don't have wisdom teeth. And for most people who do have wisdom teeth, we wind up getting them taken out because they don't fit in very well. So if you do have your wisdom teeth in, you have a full set of 32 teeth in your mouth. But if you've had your wisdom teeth removed, then you have 2 on the top, 2 on the bottom. They're going to be removed, and you're going to have 28. Now, teeth are classified according to what their job is or what their function is. So we have incisors. Incisors, the job is to cut food. Think incisors, scissors, to cut the food. And then we have canines. Canines grip or tear food. So you think about it, let's say, for example, you have a piece of beef jerky and you bite down on the beef jerky and you want to pull it off. What do? You kind of turn your mouth sideways so that you get the canines into it, and then you kind of pull it off if you can't cut through it with the incisors. So canines tear or grip the food. And then we have premolars and molars, and their job is to grind food. So we have cut, tear, and grind. Here's a diagram that shows the organization of deciduous and permanent teeth. So here we can see on this diagram, we have the location of the incisors. You have 2 incisors. You have a central incisor and a lateral incisor on both sides. The central and lateral incisors usually come in. The central incisors about 6 to 8 months. The lateral incisors usually erupt around 8 to 10 months. Then you have the canine teeth, or sometimes referred to in babies as the eye tooth. They usually erupt around 16 to 20 months. And then you have the molars. You have first molars, which come in at about 10 to 15 months, and then the second molars at about 2 years. Okay, so on each side, you're going to have 1 central incisor, 1 lateral incisor, a canine, and 2 molars, the first and second molars. In the permanent teeth, again, we have the same basic organization. We have central incisors. The central incisors are usually going to erupt around the age of 7. And then we have lateral incisors. They usually erupt around the age of 8. The canine or eye teeth usually come in at around the age of 11. And then we have the premolars, sometimes referred to as bicuspids because they have 2 cusps or lumps on them. And of those, we have the first premolar, which usually erupts around the age of 11, and then a second premolar, which usually erupts around the ages of 12 to 13. And then we have our molars. We have first, second, and third molars, and the times at which they erupt. The third molars are referred to as the wisdom teeth, and they can erupt anywhere between 17 and 25. Okay. So basically what we have then is on either side of the top or bottom, we have 1 central incisor, 1 lateral incisor, 1 canine, 2 premolars, and 3 molars. So we say dentition is sometimes referred to as human dentition as 2, 1, 2, 3. 2, 1, 2, 3, indicating the different classifications of teeth on either side. Okay. All right, let's move ahead to the pancreas, or our next accessory digestive organ. The pancreas produces a wide spectrum of digestive enzymes. As a matter of fact, most of the enzymes that are going to be released into the small intestine for the digestion of food are going to be produced by the pancreas. And these digestive enzymes break down all different categories of food. The enzymes, as we mentioned already, are going to be secreted into the duodenum, and they're secreted into the duodenum with an alkaline fluid, pancreatic juice, that has enzymes that also neutralize and the alkaline, I guess I should say, the alkaline fluid is introduced with the enzymes. The alkaline fluid is important because it's going to neutralize the acidic chyme that's coming out of the stomach. So the chyme, that liquefied uh, kind of digested food that's coming out of the stomach now, partially digested food, is from all the hydrochloric acid being secreted into the stomach, is going to be very, very low in pH. Now, the stomach itself, because of the mucous neck cells, has a thicker layer of mucus lining the lumen. So that low pH, high hydrochloric acid chyme in the stomach doesn't damage the walls of the stomach. But when it comes out into the small intestine, we don't have a thick layer of mucus anymore. So what's going to happen is that that chyme, being very low pH, could cause damage to the lining of the internal lining of the duodenum. So to prevent that from happening, as soon as the chyme comes out, it's going to mix with the juices from the pancreas, which are very alkaline, and that's going to neutralize the acetic chyme, bring the pH back up to near 7, so that we don't cause damage to the lining of the duodenum. In addition, we also have some endocrine products of the pancreas, and we'll talk more about these whenever we get into the metabolism PowerPoint. These endocrine products will include insulin and glucagons. We'll leave it at that for this PowerPoint, though. So structurally, whenever we look at the pancreas, the size, it's an oblong gland, as we can see here. It's about 5 inches long, and it's only about an inch thick. So it's not very thick at all. It's located, as we can see here in our diagram, behind the greater curvature of the stomach, and it runs from the curve of the duodenum to the spleen. So remember, here the duodenum kind of curves around, and the pancreas is going to run from here back toward the spleen here. And this is showing that it's running behind the stomach. So the stomach is kind of translucent here, and we're trying to indicate that this is an area behind the stomach. The pancreas, because it has this enlarged area, has what we call a head, and that's at the duodenal end. It has a body, which is the main portion, and then it kind of comes down, it kind of thins down to this pointed area, which is referred to as the tail, and the tail of the pancreas is located on the splenic end. Inside the pancreas, we're going to see, if we were to do a cross section of the pancreas and look at the internal structure, we would see these structures which are called acini. These acini are grape-like clusters of cells that produce and secrete the pancreatic juice. So they are cells of exocrine function. Exocrine means ducted glands. So here we can see, over here we can see the acini, okay? Those are the grape-like clusters of cells. They produce the pancreatic juice. Now, what is pancreatic juice? Pancreatic juice is a clear colorless liquid that contains water. It contains salts or ionic compounds. It contains bicarbonate ions, and these bicarbonate ions are what are going to be able to, they are OH ions. They're going to be able to combine, yes, with hydrogen ions to neutralize the hydrochloric acid or the low pH of the stomach. The pancreatic juice also contains digestive enzymes. Because of the bicarbonate ions, the pancreatic juice has an alkaline pH. So the pancreas produces the enzymes in the pancreatic juice, and it is going to be released into the small intestine. It's a ducted gland. So the pancreatic juice is going to be released into the small intestine primarily by the pancreatic duct. The pancreatic duct is going to combine with the common bile duct, as we've seen in our previous diagram, and it's going to enter the duodenum at the ampulla of Vater. Excuse me, the pancreatic duct, by the way, is also known as the duct of Wirsung, named for the anatomist who first described it. The pancreatic duct is going to extend all the way through the pancreas from the tail to the head. But in addition to the pancreatic duct, there is a smaller duct, the accessory duct. It's going to enter the duodenum separately about 1 inch above the ampulla of Vater. It will extend only through the head of the pancreas to that position just above the ampulla of Vater. The accessory duct is also referred to as, or also known as, the duct of Santorini, again, named for the anatomist who first described it. So we take a look at our diagram here again. We can see the pancreatic duct, the main pancreatic duct running the entire length of the pancreas and combining with the common bile duct at the ampulla of Vater to release pancreatic juice into the duodenum. But here we have a small accessory pancreatic duct that releases a small amount of pancreatic juice here about 1 inch above. Now what this does is, as that chyme, which has a very low pH, comes out, this secretes pancreatic juice, which mixes with the chyme and helps to bring the pH up immediately. And then additional pancreatic juice will continue to bring the pH up to around 7 as the chyme continues through this first portion of the duodenum. Okay. The liver is next. Let's talk a little bit about that. The liver is the largest gland in the body. It's a good trivia question to know. It's located on the right side of the body, just underneath the diaphragm. It consists of 4 lobes that are suspended from the diaphragm and the abdominal wall by a ligament, which is referred to as the falciform ligament. When we take a look at the liver, we look at what we call the anterior surface. This is also known as the diaphragmatic surface. The anterior surface, while the liver has 4 lobes, the anterior surface only shows 2 of the 4 lobes, and they're simply referred to as the right and left lobes. Now, next up, we have the inferior surface of the liver. This is also known as the visceral surface because it's going to face all of those visceral organs in the abdominal region. The inferior surface, or the visceral surface, has 2 additional lobes to take the total lobes to 4. These 2 additional lobes are the caudate lobe because it appears to have a tail. Caudate means tail. And the quadrate lobe because it's more rectangular in appearance. And by the way, going back to anterior and inferior surface, why wouldn't we have an anterior and posterior surface? It's because the liver doesn't hang in the abdomen kind of just this way, but it kind of is tilted a little bit. So this surface kind of faces anteriorly, but this surface under here, we could call it posterior, but it faces the visceral organs. So it's called inferior because it's below, or visceral because it's facing those visceral organs. The liver is also connected to the gallbladder via the common hepatic duct. Okay, so here are the lobes of the liver depicted in this diagram. So here we can see this only shows 2 lobes. So this is the anterior surface, and here we have the right and left lobes. And again, this is the patient's right and left lobes. And then between the 2 lobes runs this ligament, which usually the liver hangs from, but it's been removed from the body wall. So this kind of stretches up and this is the falciform ligament. If we look at the underside of the liver, the inferior surface, the visceral surface, we can see the left lobe here, the right lobe here, and we see these two additional lobes. Here we can see the caudate lobe, and if I kind of outline the caudate lobe, you can see it kind of looks like a tadpole to me. But that tadpole then has a tail, and you can see the tail kind of right there. Whoops, let's go back. I apologize for that again. Sometimes I hit the clicker inadvertently. So right there, that's that tail-like region. So that's why this gets the name caudate lobe. And then we take a look at the quadrate lobe, and the quadrate lobe looks roughly rectangular. And that's why it's referred to as the quadrate lobe. Here you can see the sac of the gallbladder and its position in relationship to the rest of the liver. And this would be the cystic duct running from the gallbladder. Okay, whoops. So what about functions of the liver? Well, the liver has more than 50 functions. And many of these functions we're going to talk about, we'll list them here, but many of them we're going to talk about whenever we go through the metabolism PowerPoint. Production of bile. Bile is important because it's going to emulsify fat or break fat apart as it enters into the small intestine. It also produces the major blood proteins. Think of the ones we talked about, um, the albumins, the proteins that are involved in the blood clotting process, fibrinogen, so many of those major proteins, not the immunoglobulins, remember those are antibodies produced by the B cells, but many of the major blood proteins are going to be produced by the liver. The liver also produces cholesterol, which is so important for cell structure and function. And it also produces lipoproteins that we'll be talking about in the next PowerPoint as well. Liver is responsible for carbohydrate metabolism, amino acid synthesis, deamination and transamination of amino acids, and those are all, again, going to be important when we talk about metabolism, so we'll leave those for that. The liver also processes hemoglobin, and it breaks down the hemoglobin and retains the iron from the hemoglobin to make more hemoglobin. So when old red blood cells die, hemoglobin is transported to the liver where it's broken down, and the iron part of the hemoglobin is retained. The liver is also important in fat metabolism. It produces urea, and it's also important in the breakdown and removal of drugs from the blood. So we'll be talking about these whenever we get into fat metabolism, we get into nutrition, and the production of urea and the breakdown and removal of drugs from the blood whenever we talk about the urinary system. The liver also stores many of the fat-soluble vitamins like A, D, K, and B12. I'll look at bile. So we know that the gallbladder stores bile, but actually bile is not produced by the gallbladder. It only stores excess bile. Bile is actually produced by cells in the liver. And bile contains bile salts. Bile salts are ionic compounds that are attached to bile acids. And that gets a little bit complicated, but so we're not going to get into that in great detail. But these salts are ionized pretty easily, and they help with the excretion of cholesterol, excess cholesterol from the body. There's also pigment. That pigment is mostly bilirubin, and that bilirubin is produced from the breakdown of hemoglobin. So whenever you take a look at bile, it kind of has that kind of bluish green coloration. That's from the bilirubin. There is also cholesterol in bile, phospholipids in bile, and that comes from the breakdown of the cells, red blood cells. When they're broken down, these components are transported to the liver. And then there are also electrolytes, which are ions that we find in the bile as well. The gallbladder itself is a sac found in the hollow fossa of the liver. There we can see the gallbladder. It stores bile from the liver by way of the cystic duct. Bile is introduced into the duodenum in the presence of fatty food. And gallstones can cause blockages in the gallbladder. Okay, so before we get on to processes, let me just talk a little bit about this with the gallbladder. And if I could go back to this, here we go. All right, so when you take a look at this slide here, see what's going to happen is the liver is going to produce the bile. And the bile is released from the liver via these two hepatic ducts. So you have a right and a left hepatic duct. The bile then passes into the common bile duct and is delivered into the small intestine at the ampulla of Vater. Excess bile is moved, that is produced, is moved back into the gallbladder, and the excess bile is stored by the gallbladder. So the next time that we eat a food that has a high concentration of fat in it, these two ducts will start to produce the bile. But in addition, the gallbladder will also contract to squeeze bile down through the cystic duct and into the common bile duct to deliver a very quick supply of bile into there and to make sure we get enough bile delivered into there. But what happens whenever a person has a cholecystectomy or whenever they have their gallbladder removed? Does that mean that they're going to have trouble emulsifying or breaking down the fat into smaller pieces to make it easier to digest? Well, they may initially, but what will happen is if the gallbladder is removed, these two ducts right here, the hepatic ducts, will dilate. In other words, they'll get larger, and the liver will compensate for that by producing additional amounts of bile and letting the bile drain down into the small intestine. Now, so a person may have trouble digesting high-fat diet for about, let's say, the first year. But after about a year, it'll adapt by dilating these two ducts and produce additional bile to help with the digestion of that food. So even if a person has their gallbladder removed, it doesn't mean they won't be able to digest fats. Okay, I'm not going to go all the way back to that next slide. The next slide is on processes of digestion. And so what we're going to do is we're going to stop there for this PowerPoint. And in our second or our first lecture here on the digestive system, and in our second lecture on the digestive system, we'll pick up with processes.